Coya Therapeutics is a clinical stage biotechnology company developing biologics and cell therapies intended to enhance the function of Tregs. And they just announced licensing of the exclusive worldwide rights of a proprietary Exosome Engineering Technology from Carnegie Mellon University with potential applications across various indications, including neurodegenerative, autoimmune, also oncology. And with me is the CEO, Howard Berman, to explain the significance of this. So explain how Coya Therapeutics and the licensing of this technology from Carnegie Mellon, how can that advance a company and be used for treatments?
Well, yes, thank you so much for having me. First, it's important to know what exosomes are. They are these little vesicles, or they're the contents of the cells that provide the information that permits the cells to do what they need to do. So when cells act in the body, they release these exosomes, and they are very potent. What we've been able to do is to be able to isolate these exosomes, and using technology from Carnegie Mellon is be able to modify the exosomes so that now you can actually load these exosomes with cargo, drugs, and proteins, or even messenger RNA, or you can create potential vaccines in the interior of the exosomes. And then on the surface of the exosomes, we can target them to specific tissues or proteins. So you can selectively guide them in a very targeted manner. This really changes the way that this can be modified. And traditionally and previously, it's been very complicated to do so. Lots of complex molecular biology techniques, and it's just not been a rapid and scalable manner to do so. And this Carnegie Mellon technology allows us to overcome those issues.
Wow, that's really interesting. So this will allow you to overcome some of the limitations that you've had before, this particular technology, and it will enable you to target drug delivery? Is that kind of the takeaway here?
Right. Some people call it the new drug delivery platform, because instead of using, some people have used liposomes and other carriers, this is a very biologically compatible methodology that you can now essentially load whatever you want into these exosomes and have it distributed around the body, because these exosomes are very small and they can penetrate the blood-brain barrier and they can penetrate tissues. So it's really a next generation delivery technology that opens up tremendous opportunities for collaboration and partnership and strategic opportunities with pharma companies.
Wow. So can you provide a little more details about the successful engineering of the exosome with proteins like CTLA4, FASL, and how that can selectively target immune cells?
Correct. You can modify the outside of the exosomes, and we've done so with CTLA4. What that allows the exosomes to do is to target cells that are inflammatory cells. And so you can selectively target conditions that are driven by inflammation. So that's one thing that we've accomplished recently. We've also been able to leverage the data from Carnegie Mellon, where they've targeted FAS protein, which is an apoptotic protein, and been able to upregulate that and cause apoptosis in cancer cells. So now you can actually target the complete opposite. Instead of inflammatory diseases, you can target cancer-based disorders. And then on the interior of the cell, we've shown how you can put growth factors on the interior and deliver those growth factors to cells of interest, whereas the growth factors haven't been able to be delivered as such before, because you need such high concentrations if you're going to deliver them. So it overcomes many challenges by loading the exosomes and also targeting them to proteins of interest.
So how does the expansion then of Coya's pipeline beyond neurodegenerative diseases into, it sounds like cancer, also autoimmune, how does that fit with your long-term strategic goals?
Sure. So we are a company focused on neurodegeneration initially and inflammatory-based disorders, but we also know and value that the technologies that we have expand us into oncology and other conditions. While we want to be focused on the core assets, which are our ALS and our biologics, we also realize that the technologies can be leveraged by pharma companies. And we are in numerous discussions with multiple organizations and companies to advance our exosomes and some of our programs there. So we want to use some of this exciting data to prop up or leverage their capabilities with the advantages that we can provide with our technologies. And there's lots of examples that we can share in the future.
Well, that's what I was just going to ask is what's next then for this exosome engineering? Now that you've got this data, what happens now?
Well, just imagine if we could show proof of concept data, for instance, that you could create a better vaccine, you could put mRNAs inside the exosomes and deliver it in a better way, or you could put specific molecules and drugs and target those to certain cancers by targeting specific proteins. I think you would have a very strong case for companies, big companies, who are suffering issues in terms of delivery, that they may find this of an interesting opportunity to collaborate.
Okay, well, I would look forward to updates from you as this progresses. Thank you so much, Howard, for joining and explaining this significant event.
Thank you. A pleasure of mine.